Welcome to Computational Biology. Today, we are diving into sequence alignment, the absolute foundation of understanding how life evolves and functions at a molecular level. If you have ever wondered how scientists trace the tree of life or discover cures for genetic disorders, it all starts right here. Let us begin our journey. To understand what sequence alignment is, imagine you have two different editions of the same book. You want to find out exactly what sentences were added, deleted, or changed between them. You would place the texts side by side and match up the identical words, leaving blank spaces where a word is missing in one version. In computational biology, sequence alignment is exactly like this. But instead of words in a book, we are comparing the individual letters that make up the code of life. Now that we have our book analogy, let us apply this to biology. Instead of English sentences, we are comparing the individual letters of DNA, RNA, or proteins. Formally, sequence alignment is the process of arranging two or more of these biological sequences to identify regions of similarity. When we align them, we often insert gaps, which are usually represented by dashes, into one or both sequences. The ultimate goal here is to maximize the number of matching characters while minimizing the number of gaps and mismatches. You might be wondering, why does this simple act of lining things up matter so much in the real world? It is actually the primary tool scientists use to discover the function of a newly sequenced gene. For instance, if a new gene aligns perfectly with a known gene that causes a specific disease in mice, we can hypothesize that the new gene might cause a similar disease in humans. Without this technique, massive databases of genomic data would be completely unreadable. It is the engine driving drug discovery and personalized medicine. Knowing why it is important leads us to a critical question. How do we decide if an alignment is good or bad? We use a mathematical scoring system, much like grading a multiple choice test. You get points for a correct answer and lose points for wrong answers or skipped questions. In our formula, S represents the total alignment score. The N variables represent the count of matching characters, mismatched characters, and gaps. The P variables represent the point values assigned to each. Matches get a positive score, while mismatches and gaps receive negative penalties. If we increase the gap penalty, the computer tries harder to avoid inserting gaps. Let us put this formula into practice with a concrete example. We want to calculate the alignment score for two short DNA sequences, ATCG and ATCC. We are given a simple scoring system. A match gives positive 2 points, a mismatch gives a negative 1 point penalty, and a gap gives a negative 2 point penalty. Our goal is to find the total score S if we align them directly without any gaps. This is a very common basic scenario in sequence analysis, and we will solve it by comparing the sequences letter by letter from left to right. Now let us count our variables and plug them into the formula. Looking at the sequences, we find 3 matches. The first 3 letters, A, T, and C. So we multiply 3 by the match reward of 2. We found 1 mismatch at the end, G versus C, so we multiply 1 by the mismatch penalty of negative 1. There are 0 gaps, so that part of the equation becomes 0. Summing these values together, 6 minus 1 plus 0 gives us a final alignment score of 5. This positive score indicates that the two sequences are highly similar. We just calculated a score of 5, But what does that actually mean in biology? A high positive score like this suggests that these two short DNA fragments likely share a common evolutionary ancestor. Because 3 out of 4 letters are identical, it gives us confidence that they might perform a similar function in the cell. This is exactly how researchers use computational tools to predict the behavior of unknown genetic sequences based on known ones. Now let us look at a slightly more complex scenario involving gaps. We have 2 new sequences, AT-G and ATCG. We will use the exact same scoring system as before, +2 for a match, -1 for a mismatch, and -2 for a gap. Evaluating column by column, we have 3 matches, A, T, and G. There are 0 direct mismatches. However, we have 1 gap, represented by the dash in the top sequence. We need to calculate the new total score to see how this gap affects the overall alignment quality. Let us substitute our new counts into the scoring formula. We multiply our 3 matches by the match score of 2, giving us 6. The mismatch term is 0. For the gap, we multiply 1 by the gap penalty of -2. Adding these components together, 6 plus 0 minus 2 gives us a final alignment score of 4. Notice that this score is slightly lower than our first example, even though there are no direct mismatches here. The gap has reduced the overall quality score of the alignment. This lower score of 4 teaches us a very important biological principle. Why did the score drop? In computational biology, introducing gaps is heavily penalized because such mutations are generally rare and disruptive in nature. Gaps represent actual physical insertions or deletions in the DNA sequence. These physical changes can drastically alter the shape and function of a biological molecule, often much more than a simple single-letter mismatch. Therefore, our mathematical scoring system is designed to reflect this biological reality by making gaps costly. To fully solidify these concepts, let us look at a visual diagram of a DNA sequence alignment. Here, you can clearly see the top sequence and the bottom sequence lined up. The vertical black lines indicate perfect matches, where the letters are identical, like the A and T at the beginning. In the middle, you will notice a red cross. This highlights a mismatch, where a C aligns with a G. Further to the right, a blue arrow points to a gap, represented by a dash in the bottom sequence. This diagram perfectly illustrates how algorithms physically arrange sequences to identify these 3 key features. We have covered a lot of ground today, so let us summarize the key takeaways. Sequence alignment is the fundamental computational method for comparing biological sequences to find regions of similarity. By assigning mathematical scores to matches, mismatches, and gaps, we can objectively measure how closely related 2 sequences are. This scoring system allows computers to search massive databases automatically. Ultimately, these alignments are what help scientists decode evolutionary history, predict protein functions, and identify the genetic causes of diseases. You now understand the core engine of computational biology.